drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. This episode is ad-free, and I rely on my audience to support my efforts. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode.
My pleasure. It's fun to be here.
For those who don't already know you and your work, can you give us an idea for what you study in your lab and what you've studied over the course of your career?
Well, I'm a neuroscientist and a psychiatrist who has spent my career at institutions in the Bay Area, at Stanford, then a place called UCSF, then back to Stanford. And my lab, initially, I've had a lab for a long time, for 35 years. My lab initially focused on a topic that we call synaptic plasticity, which is a catch-all term that encompasses how synapses, the connections between nerve cells, how they change in response to experience. And that experience can be a form of learning and memory. That experience can be administration of a drug or the experience of a drug, a stressful event. And most neuroscientists believe that part of the way we encode experiences and remember them and their impact on our brains, and therefore how our brains adapt to experiences and change our subsequent thoughts, feelings, and behavior. Most of us believe that changes in the properties of synapses play a very critical, important role. So really, probably for the first 20, 25 years of my career, a lot of my work focused on how do these changes in synapses in response to changes in the patterns of activity in the brain, how does that actually happen? Which proteins at the synapse, which receptors, and I apologize, I'm not sure of who's listening to this, so I don't know if I need to define terms such as receptors, but the proteins in the membranes of nerve cells that respond to chemical messengers that we call neurotransmitters. Anyhow, how these changes in synapses actually happen at a fairly molecular level. And then that led me to start doing work in the behavioral relevance of these various forms of synaptic plasticity. That is, I'm studying synapses and how they change in very reduced systems. And I became very interested in, okay, if this is really important, I should be able to correlate these changes with behavioral changes in my favorite experimental species, which is a mouse or mice. And then gradually, over the decades, while I continued to study the molecular mechanisms of synaptic plasticity, that led me into all sorts of different research topics including how a neuromodulator we call dopamine, how it works, what it does in the brain, studying changes in what we call the reward circuitry in the brain, which my guess is we're going to end up talking about in the next hour, hour and a half. That led me to studying another neuromodulator that goes by the name of serotonin and some of its important functions in the same reward circuitry. And then finally, that led me to studying certain psychedelic drugs. But there was actually a natural progression of how my research led me from studying pretty molecular mechanisms to the behavioral effects of psychedelics. There's actually a thread throughout my scientific career.
Interesting. Yeah. And hopefully we sort of trace that thread to some extent. And I think we will. When you were just getting started, back in the early days of synaptic plasticity and some of the early discoveries of the initial mechanisms that we discovered, when we think about things like NMDA receptors and long-term potentiation, and we can briefly define those things, but I've talked about them on the podcast before. A lot of people listening will have some familiarity with those terms. When those things were being discovered, the mechanism through NMDA receptors by which, this coincidence detection, synaptic strengthening happens, what was the thinking like leading up to that? Did people have a notion for how that would work, or was it completely surprising? What was that initial sort of set of discoveries like?
It's a fun scientific story. So, it sounds like your listeners and you certainly know that the major form of plasticity that I have studied and others in the field have studied is called long-term potentiation, or LTP, which is shorthand for long-term potentiation of synaptic transmission. It's been studied primarily in the region of the brain called the hippocampus, which made a lot of sense because we know the hippocampus is critical for the storage of memories, the storage of new information. It was also studied in the hippocampus in terms of trying to understand the underlying mechanisms of LTP. We use the hippocampus because we could cut slices of it and put them in a dish, and it offered a lot of experimental advantages. And so the big breakthroughs in LTP were sort of a natural progression. Some of the basic properties were elucidated initially in some classic papers by some European neuroscientists. And then a critical of observation was, as you alluded to, the finding that NMDA receptors, this subtype of receptor for the excitatory neurotransmitter glutamate, was critically required for the initiation or triggering of the biochemical events that lead to LTP. And what was really interesting about that discovery, and it was a very simple experiment, just bath applying an NMDA receptor antagonist to a hippocampal slice. And the key finding there was that this antagonist to NMDA receptors blocked LTP, but it did not seem to affect what we call basal synaptic transmission, or basal synaptic strength. And that was, at the time, pretty bewildering. So how could a drug have this, a blocker of NMDA receptors have this very clear effect on LTP, but have no detectable effect on basal synaptic transmission? And that remained a mystery. It's been so long, I can't remember how many years it took. And then two different groups started studying NMDA receptors at a biophysical level, and they made the very important discovery, and I hope this isn't too much scientific detail, that the NMDA receptor was a very unusual ligand-gated ion channel that responds to the neurotransmitter glutamate because it was voltage dependent. That is, when glutamate bound to the NMDA receptor, when the cell was just hanging out at what we call its resting membrane potential, the NMDA receptor didn't pass current, didn't pass very many ions. And it turns out that's due to the fact, and again, this may be too much detail, that a divalent cation called magnesium sits in the pore of the NMDA receptor and it actually isn't quite true that no current or ions can flow in and out of the NMDA receptor channel, but it's pretty much blocked. But when the cell gets depolarized, gets activated by other synaptic inputs or for whatever reason, the magnesium is expelled out of the ion channel. And now when glutamate binds to the NMDA receptor, ions can flow, currents are generated, and the cell can respond. And most important, so there was a voltage dependence to the response of the NMDA receptor. And then the other key finding from two different groups was that unlike many other glutamate receptor channels, the NMDA receptor was permeable to calcium. So when the cell was depolarized, calcium and activated by glutamate, calcium could enter the cell. And so those biophysical findings, let me just remember my history here, were made in the 80s. Mm-hmm. And two different groups. The group I was involved in when I was a postdoc with Roger Nickel and a guy named Gary Lynch, sort of looked at these papers and realized, well, if what makes the NMDA receptor so special is that calcium can enter through its ion channel, maybe calcium is a critical trigger for LTP, but the calcium entering through the NMDA receptor. So then Gary Lynch's lab did an experiment that very simple in today's world, but back then it was a very clever experiment. I hope I'm remembering this correctly, where he recorded from individual hippocampal cells and loaded the cells with a compound that sucks up the calcium. It's called a calcium chelator and prevents the calcium from binding to other proteins and doing its magic. And if I'm remembering correctly, that blocked LTP. And so that was, although the finding actually didn't get as much attention as maybe it should have. And then work I did with another postdoc in Roger Nickel's lab, Julie Cower in Roger Nickel's lab, and this was work done at UCSF. We showed not only that we replicated that observation, that if we chelated the calcium by loading the postsynaptic cell from which we were recording synaptic responses, which are called EPSPs, excitatory postsynaptic potentials, we could block the generation of LTP. And then very importantly, we showed if we used some very cool molecules that we could load into the cell and they held on to the calcium. But when we flashed a light, calcium was released in the cell. We showed that could cause an LTP-like effect. So we had both necessity, meaning if we blocked chelated the calcium, prevented it from doing its thing, we blocked the generation of LTP. That shows necessity. Then we showed sufficiency by showing if we just released the calcium, independent of activating NMDA receptors, we could cause an LTP-like effect. We could strengthen synaptic transmission. So that then really, I think, opened up the field because now there was a very clear explanation for the initial triggering events of LTP and why it required depolarization of the cell. And in the real brain, we think that happens by the coordinated activity of different inputs onto that cell, which starts depolarizing the cell, making it more excitable. So then when another synapse is activated in a temporally coincident manner, the cell's already depolarized. The glutamate that is released from that synapse activates the NMDA receptor. Because the cell is depolarized, calcium can enter that specific synapse, trigger a cascade of biochemical events that lead to LTP. And so everything came together in a very beautiful scientific, it's how science should work. When, it took, the truth is, LTP was first described, I hope I'm remembering this right, in 1973. And it really took 15 years till the late 80s to elucidate what is really a relatively simple mechanism.
Mm-hmm.
It's a combination of the initial description of LTP, the discovery of NMDA receptors and their role in LTP, the description of their biophysical properties, and then labs like the work I did in Roger Nichols' lab with Julie Kaur and Gary, putting it all together and saying, testing the hypothesis that LTP was triggered by depolarization, activation of MDA receptors, and the entry of calcium. And then that led, if we're, to all sorts of additional experiments. Yeah. Probably more than you needed to know.
No, I mean, it's a fascinating story. And, for those who don't know, like the mechanism is sort of beautifully simple. It's probably one of the most beloved mechanisms in the brain that
it's now in textbooks. It's now just standard textbook knowledge.
But if I sort of reiterate some of what you said, we've got this sort of special receptor, this protein complex, the NMDA receptor, and it doesn't really pass much current through under normal baseline conditions. But if a cell becomes excited enough because, say, multiple neurons are simultaneously activating it, it can suddenly pass more current. And there's a couple of interesting ions involved here. On the one hand, you've got this magnesium. So like when we eat magnesium in our food, some of this magnesium is being used to block this channel so that nothing is really going through it. But then when the neuron becomes sufficiently excited, now calcium can go through it and get inside the cell. And calcium, most people probably know calcium when they think about their bones and their teeth, but it's important in neurons, it sounds like, for actually getting into the cell to trigger the things inside the cell that are necessary for strengthening these synapses.
That's exactly correct. Very well put. Although, as we may get to, calcium can also decrease synaptic strength and cause the opposite of LTP. So, like everything, once you start delving into the details, the biological processes involved get more complicated and more flexible.
Mm-hmm. And so long-term potentiation, that's what we just described. That's at least one way that synapses can get stronger.
Right.
Are there is that the primary mechanism, or are there like a whole bunch of ways you can strengthen a synapse that work in
strengthen? It's a very good question. I have to go through the Rolodex in my head. It turns out that in some cells, including the hippocampal cells that are the main source of the scientific information we have about how synapses change, you can actually, if you depolarize the cell repetitively with action potentials, you can probably load the cell with calcium via what are known as voltage-dependent calcium channels, which is just another source of calcium that when these calcium channels are repetitively activated to a sufficient degree, that can lead to the strengthening of synapses. Now, whether that actually ever happens in the awake behaving brain remains unclear. Let me just think. Are there other examples? Yeah. It turns out at certain synapses, there's a very different way of strengthening synaptic transmission. So what we just talked, and then I hope we're going to talk about the weakening of synapses, which could be as important depending on the circuit in which those synapses are embedded. But at certain sets of synapses in the brain, which are embedded in certain circuits in specific brain regions, there's a form of LTP that, rather than being triggered by postsynaptic activation of NMDA receptors, is triggered presynaptically due to certain repetitive activation of the presynaptic terminals that release the glutamate. And, historically, that was first described at a set of synapses in the hippocampus called mossy fiber synapses. And so it's a very different form of synaptic strengthening called mossy fiber LTP. Interestingly, it also involves the key triggering mechanism is the entry of very high amounts of calcium in the presynaptic terminal through these voltage-dependent calcium channels. But I think most of us in the field believe NMDA receptor dependent LTP, which we were talking about previously, which is what I would call the prototypic form of LTP and synaptic plasticity. It occurs at many different excitatory glutamatergic synapses throughout the mammalian brain.
I see.
You
So it's
find
pretty
it
common.
all
Different
over the
circuits,
brain.
different brain regions.
Exactly. This other form I was describing, which is this presynaptic form of LTP, we call mossy fiber LTP. I mean, it's only so far, it's only been found at a very small number of kind of specialized synaptic inputs in the hippocampus at the mossy fiber synapse, and at a certain synapse in a region of the brain called the cerebellum, which is important for motor movement, for controlling movement, and maybe one or two others that I'm not just remembering at the moment. Mm-hmm. And then beyond that, beyond NMDA receptor dependent LTP, mossy fiber, I'm sure when we're off this, I'll think of one or two others, but there's not a lot.
I see. So the sort of classic NMDA receptor LTP is a major way that synapses are strengthened across the brain.
I think most neuroscientists would agree with that. Absolutely, and that's why, at least historically, it's not a very popular topic currently, now that we're in 2024. But in the 70s, 80s, and 90s, it was a very popular topic to study that received a lot of attention.
And so sort of the flip side of this is as you've alluded to earlier, you not only want to strengthen synapses in the brain, you want to weaken and sometimes get rid of synapses in the brain. How does that happen?
Okay, so that phenomenon is called, I guess neuroscientists are not that imaginative. It's called long-term depression of synaptic transmission, or LTD. And it's an interesting history. Once LTP was described in the early 70s, all neuroscientists started thinking, well, it might be advantageous for several different reasons to have the opposite phenomena, that is long-term depression. Some people argued you don't want to have a permanent strengthening of synapses. You want to have a way of weakening them. Perhaps that's a mechanism of forgetting, which I actually am not sure it is. I think it's more complicated than that. Other people just posited that circuits within the brain would have a lot more flexibility and power if they could utilize the bidirectional control of synaptic strength. That is, if synapses mediating information processing in this or that circuit could both strengthen the synapse and weaken synapses. If you do simple-minded computational modeling, the flexibility of how that circuit functions is enhanced enormously by having both LTP and LTD. So then let me get my memory right, in the 70s and 80s, there were papers published reporting phenomenologically various forms of LTD, primarily in the hippocampus, but they never really gained traction. I think primarily because experimentally they were a little bit ephemeral. They were hard to induce. Different labs couldn't get the same reliable generation of LTD, whereas LTP, once you know what you're doing, most people, even very novices in the field, could generate LTP relatively easy, let's say in a hippocampal slice. So then in the early 90s, a colleague of mine who's now at MIT, Mark Baer, developed a very simple protocol based on some theoretical work he had been involved in for inducing LTD in the hippocampus at the same set of synapses at which LTP had been studied for decades. And then Mark continued to study this form of LTD, and then I noticed it because he was a friend and he told me about it, and I said, hey, do you mind if I work on this? And so we started studying LTD in the hippocampus, as I said, at the same set of synapses. And because we knew how to study synaptic plasticity mechanistically from all the years we had been studying LTP, it was very easy to very quickly elucidate some of the basic triggering mechanisms of this other form of plasticity, synaptic plasticity. And interestingly, what we found is that LTD was NMDA receptor dependent, just like LTP, but it required a different pattern of activity at the synapse. And the simple model we followed, which was actually first proposed by a neuroscientist named John Lissman, who was at Brandeis. Unfortunately, he passed away several years ago. And what John Lissman had proposed in a theoretical paper is that LTP involves an NMDA receptor dependent increase in calcium in the postsynaptic cell, but it had to be a very large increase in calcium beyond some critical threshold. Let's say the threshold was 10 micromolar. It had to be above that. And that perhaps LTD could be triggered if a much lower level of calcium was achieved via the NMDA receptor, maybe in the range, I'm making this up, of 1 to 2 micromolar. So we actually tested that kind of thinking and hypothesis, and all of our results were consistent with it. And to be honest, I think it's now kind of dogma in the field. I believe it's in textbooks that there is an NMDA receptor dependent LTD due to the increase in calcium in this narrow window. And then I don't know if we're going to talk about the biochemical cascades activated by calcium, but a certain set of calcium-dependent enzymes are activated by a low level of calcium that trigger LTD. And then NMDA receptor-dependent LTP requires what I said, a much higher level of threshold. And then that led to people looking for different forms of LTD all over the brain. And there are probably many more forms of synaptic depression, or LTD, than there are forms of LTP. And why that is, and how the nervous system and circuits in the brain utilize these different forms of synaptic plasticity, is still an active area of research.
So it sounds like there's a lot of ways to make synapses weaker.
Yeah. I now that I said that, I have to start going through the Rolodex in my head. And I think there are several, yes.
So why do you think that might be? Why is it so important to weaken synapses? Have we done experiments where we prevent the brain from doing LTD, and does that impact learning?
A very good question, and I haven't worked on these phenomena, oh my God, in a long time. Actually, I think one hallmark example is in the cerebellum. There's a form of LTD that it's been so long since I read these papers, that involves two different inputs onto a major cell type in the cerebellum called the Purkinje cell. And if the timing of these two synaptic inputs is appropriate, there's a weakening of the synapse between a set of axon inputs called the parallel fibers. And then there has been quite a bit of work proposing theoretically and then experimentally demonstrating that this form of LTD seems to be required and involved in certain forms of cerebellar dependent behavioral plasticity. So that's one example. I'm trying to think of other examples. It's going to take me a while to think of.
But basically, there's multiple mechanisms of synaptic plasticity. Synapses can get stronger, they can get weaker. There's a number of ways each of those can happen.
Yes.
Calcium is critical for all of this. Calcium getting into neurons is super important.
Pretty much, except there's another form of LTD which has gotten a lot of attention. And again, calcium is sort of always directly or indirectly involved, but this is a very interesting form that involves oh God, it gets a little complicated. The release of these endogenous brain modulators called endocannabinoids. And I don't know if we want to get into that, but this is the brain's natural marijuana, or its natural THC. And it turns out that in certain cells and at certain synapses, strong repetitive postsynaptic depolarization of cells or spiking of cells can, in a calcium dependent way, so calcium is still involved, cause the release of these substances called endocannabinoids that then leave the postsynaptic cell and then attach to receptors on what we call the presynaptic terminals and reduce the amount of glutamate or neurotransmitter that is released. That's called endocannabinoid mediated LTD. And that's seen that's seen in the hippocampus, that's seen in a part of the brain known as the basal ganglia or the striatum. And there's some evidence that it's important for certain types of behavioral phenomena or behavioral plasticity.
I guess would the basic idea here be that, if a neuron is getting too much input, it sort of sends an endocannabinoid backwards to the cells talking to it and says, quiet down a little bit.
I think that's a nice way of putting it, a very nice way. And again, I think describing these synaptic plasticity phenomena, has been very important, and people have studied them in many different brain regions, in the hippocampus initially, in the cerebellum, in various cortical areas. A larger experimental challenge, which people have been taking on and working on for decades, is how are these synaptic plasticity phenomena actually used in the intact, awake behaving brain to mediate all the wonderful plasticity that happens as we experience the world, or as our mice experience the world. And there's hundreds, if not thousands, of papers on this topic.
Is when we think about neuroplasticity and the brain's ability to change itself in response to experience, is it all about synaptic plasticity, or are there other forms of plasticity that exist outside of the synapses changing?
It's a very good question. And I think there are certainly other forms of plasticity. I think most neuroscientists would agree that synaptic plasticity is a major contributor to neuroplasticity. I mean, the term neuroplasticity, especially among lay people, and especially among certain cadres of neuroscientists, is used very loosely, because it basically means any change in the brain is a neuroplasticity phenomena, anything. So, but to answer your question, so synaptic plasticity is certainly really important, but it is very likely that there are forms of plasticity that involve longer lasting changes in what we call the intrinsic excitability of cells. So, cells have voltage dependent channels that generate action potentials or spiking in the cells. And there is evidence that, again, in response to certain patterns of input activity, the cell can change, or in response to certain neuromodulators, the cell can change its spiking behavior. That is, for a given amount of input into the cell, it may spike more, it may spike less. And that could be a very powerful form of neuroplasticity. And then there's forms of neuroplasticity, which are probably connected to synaptic plasticity, where new synapses, new connections can be formed. So that's structural plasticity. And synapses can be taken away, they can disappear. And then most of us in the field believe that these forms of structural plasticity the formation of new synapses, the disappearance of existing synapses, are probably to a large extent triggered by LTP and LTD. So, and there's actually experimental evidence pretty strong to support that proposition.
Mm-hmm.
And let me just think. And then it sort of depends on how you define neuroplasticity, because there are long-lasting changes in gene expression. And that's a form of neuroplasticity. What I have always pointed out, and I don't want to say argued, just pointed out, that when there are long-lasting changes in transcriptional regulation and epigenetic changes and transcriptional changes, they still have to, in order to affect neural circuit function, neural activity, they still have to eventually influence something about the physiology of the circuits. They have to eventually change the spiking behavior of the cell, or how synaptic inputs are influencing that cell. Mm-hmm. Otherwise, they're not really having they're not influencing the functions of the brain. That is, our thoughts, our feelings, and behavior. So I hope I'm answering your question. So there are many different forms of neuroplasticity.
Mm-hmm. And there's also this concept of metaplasticity that others have discussed. We might loop back to this when we talk about things like drug addiction and psychedelics, but what's metaplasticity as opposed to just plasticity?
It's been so long. I mean, metaplasticity is the plasticity of plasticity mechanisms. So
how easily a cell can you use to deploy those mechanisms?
Yeah, I mean, so I have to remember. So it can be, for example, we talked about the triggering mechanisms for NMDA receptor dependent LTP and LTD. Metaplasticity would be in response to some experience that the actual patterns of activity that trigger LTP and LTD have changed. So the mechanisms of LTP and LTD may still be the same, but the patterns of activity, because of changes in the circuit, have changed. It also can be that the biochemical mechanisms triggering NMDA receptor dependent LTP and LTD have been modified over the course of days or weeks. So it's really the plasticity of plasticity. And it even can be that additional mechanisms that before this behavioral experience that triggered LTP, that the mechanisms have been modified somewhat. So you need additional activation of a different subtype of receptor or something like that. Mm-hmm. So it's a term that is sort of generally used, as I said, to say it's the plasticity of plasticity mechanisms.
And so an area where you've done a lot of work on synaptic plasticity is in the so-called reward circuitry of the brain, the mesolimbic dopamine reward system, which is very important for learning and memory and responding to natural rewards. And it's also important for drug addiction and the response to various drugs of abuse. I want to talk about that stuff a little bit. Very briefly and concisely, can you just give your sort of definition or perspective on what actually counts as a drug addiction or an addiction behaviorally?
Well, I mean, clinically, the definition is the continued pursuit and ingestion of a substance, and this is the key phrase, despite severe adverse consequences. So people often ask me, can I be addicted to coffee? Well, not by that definition. You never hear about people robbing, their family members, committing a crime, a marriage breaking up because they need that cup of coffee. And I mean, so you can have a strong, and so people use it loosely, but the medical, diagnostic definition is what I said: the pursuit and ingestion despite severe adverse consequences.
So it sounds like there's a difference, and it might be a matter of degree, but there is a difference between really wanting something and compulsively seeking it out.
That's exactly right. And again, like many diagnoses, especially in the psychiatry realm and mental illness, there's a continuum. But yeah, exactly. It's this compulsive seeking despite adverse consequences. But, I'm very grumpy in the morning if I don't get my coffee, but I would not,
You're not going to rob a bank to get it.
Addict by that definition.
Yeah. Yeah, I talked to Christian Lucier, and he made the distinction between dependency, which is, you're waking up with a headache and being grumpy in the absence of the caffeine, and the compulsion side of it. He basically said you have to have both to qualify as addiction in the clinical sense.
I mean, although you can be, yes, basically dependency goes along, but I think Christian's point is very well made and well taken. I'm dependent. Yeah, I'm a little bit, and you can have withdrawal symptoms without being addicted. People talk about if they don't get they can get headaches if they don't get their caffeine because they're so used to it. But that doesn't mean they don't meet the medical definition of addiction.
And so there are many addictive substances out there. I'm sure we'll maybe use some as examples: nicotine, opioids, amphetamines. The list goes on and on. To what extent are they all acting through unique mechanisms, and to what extent is there sort of a common core element in the brain and what's going on in terms of plasticity that they all share in common?
Yep. I mean, so, a big breakthrough in our understanding of the neurobiology of addiction was the realization and I'm forgetting, let me just think, I think in the 80s, that it seemed that all these different drugs with the term I like to use is addictive liability. So drugs, have different addictive liability. So, a drug with very high addictive liability is fentanyl or opioids, psychostimulants like cocaine, methamphetamine. Other drugs may have slightly less addictive liability, but they all seem to cause the release of this neuromodulator dopamine in a key component of the classic reward circuitry in the brain called this horrible name of the nucleus accumbens. And I'm sure if you talk to Christian Lucier, he described this. And what's really, so that was one major breakthrough. And then the other was that these different substances, opioids versus psychostimulants like cocaine, methamphetamine versus nicotine, all cause the release of dopamine in the nucleus accumbens, but at a receptor level and at a circuit level, they do it via different mechanisms. But this realization of the key triggering mechanism that leads to addiction being this unphysiological release of dopamine really opened up the field to a more mechanistic neurobiological description of how addiction develops and is maintained.
I see. So nicotine, cocaine, all of these different drugs of abuse, they might act through different mechanisms, touch different receptor systems and things in the brain, but ultimately they're converging on elevating dopamine in certain key brain regions beyond sort of what a natural reward would do.
And that's the whole key, is it's beyond a quote natural reward. So I really like donuts. And I guarantee you when I'm hungry and I eat a donut, I get a spike of dopamine in my nucleus accumbens, but it's not even remotely to the same degree as the spike of dopamine I would get in a target region like the accumbens if I, smoked or injected fentanyl. It's a different beast. And that's, one of the major reasons we have somewhat of an addiction epidemic in our country is our brains evolved to respond to natural rewards because it was really important to have a mechanism in our brain to tell us, this is really good. I need to remember where this food source was or where this sexual partner was. So our brains evolved for natural rewards, but then, really for the modern drugs, which are things like morphine, fentanyl, psychostimulants, those didn't even exist till the 20th century, and our brains weren't ready for them. So
Mhm.
and so the connection to synaptic plasticity, to bring it full circle, was, and I will take a little credit for this, is that I was studying LTP and LTD in the hippocampus and figuring out the mechanisms, but I was interested about connecting up these forms of synaptic plasticity to some form of behavioral plasticity, because even in the 80s, it was really hard to directly demonstrate that the phenomenon of LTP in the hippocampus was in fact required for hippocampal dependent memory. So I started thinking about other brain areas to study synaptic plasticity, and I started, reading literature in all sorts of different fields. And this was probably in the late 80s, although we didn't really do the key experiments till the 90s. And people who had been studying the actions of drugs of abuse and were working on the neurobiology of addiction, they had started proposing the idea that maybe synaptic plasticity mechanisms like NMDA receptor dependent LTP, but in a different key region, in particular in an area of the brain called the ventral tegmental area, another horrible name to remember, but it's the home of the dopamine neurons. It's the home of the neurons that send projections or axons to other key components of the classic reward circuitry. So my colleagues in the field were proposing that maybe drugs of abuse triggered an LTP-like event at synapses on dopamine neurons, but nobody had ever done experiments to directly test that hypothesis. So then in the late 90s and early 2000s, with an ex-postdoc of in my lab, a guy named Antonello Bonci, we actually tested that hypothesis. So we administered drugs of abuse. We started with cocaine, and then we expanded to other drugs of abuse, and this became important. And then using some what are now standard methodologies, but at the time were a bit clever, I would say. And it's because I had a background in understanding how to study synaptic plasticity and how to measure synaptic strength in brain slices. So the experiment we did is we administered a drug to a mouse, and the first drug was cocaine, and we studied glutamatergic excitatory synapses on VTA dopamine neurons, and we made a measurement that correlated with synaptic strength, and we showed that cocaine caused an LTP-like change. And then we showed in a subsequent paper done primarily in my lab that and this was key because the question was, is this LTP-like effect on dopamine neurons, does it only occur in response to in vivo, meaning in the real mouse, administration of cocaine, or do other drugs of abuse cause the same change? And then we showed that nicotine, alcohol, I'm trying to remember, I think morphine, all caused a similar LTP in these midbrain dopamine neurons. That then opened the door for all sorts of different synaptic and circuit work on looking at how synapses and circuits that are part of the reward circuitry, how they change in response to drugs of abuse, and which changes last long enough that maybe they contribute to addictive behaviors. Probably more than you needed to know.
So no matter what drug of abuse we're talking about, they can act through very different mechanisms, but they sort of all converge by causing an unnaturally high level of dopamine release in certain key regions of the brain. Right. And that's tied into the strengthening of synapses through LTP. And that's ultimately going to be required for addiction to set in.
Yeah, I mean, the hypothesis that at least one of the initial triggers is LTP at synapses on the dopamine neurons.
Mm-hmm.
But there are other mechanisms. That's
Mm-hmm.
not the only mechanism, just to be clear.
Got it. Yeah. So there's more than one way that this dopamine release can initiate the strengthening.
Yeah, it's a combination of a number of different biochemical and synaptic changes in response to a drug of abuse that then lead to the cascade of events that we define as addiction.
And I like what you said earlier about addiction liability. So instead of thinking of drugs as being addictive or non-addictive, we should think of them as existing on a spectrum of addiction liabilities ranging from very low to very high and everything in between.
Absolutely.
When we think about something that's usually considered to be pretty addictive, like a psychostimulant, like cocaine, say, and we say that it has, a certain addiction liability, what does that mean in terms of, does everyone have the same percent chance of becoming addicted upon repeated exposure, or are some people likely to become addicted and some people sort of immune from becoming addicted? How do we think about that at the population level?
Again, I think, like everything in medicine and everything in our human behavior and how we react to our environment, it's complicated. And, I teach this topic, at Stanford University to undergraduates. And, what I always say is, you cannot become addicted to a substance if you never ingest the substance, right? By definition. So if you're thinking of trying fentanyl or trying methamphetamine, just be aware of that. If you don't take it, you can't become addicted to it. But then the truth is, there are people have different responses to these substances. And that's probably has to do with their underlying genetic makeup. So let me just give you another example from my own experience. Alcohol. If I drink a few drinks, I like to drink. I find it is a social lubricant for me. I have actually been inebriated over my life, but most of the time I get sleepy and I don't, it's not unbelievably rewarding to me. I mean, it's fun, but it's not but then you talk to certain people who really developed, what we would call, what we used to call, they became alcoholics. We now say they had an alcohol use disorder. They will tell you the first time they had a martini or a drink, it was like the best feeling in the world, best feeling they had ever had in the world. And there's something genetically different because my actual physiological response was different. And you can talk to people with the same kind of difference and continuum of responses to the use of cocaine. Some people actually don't like it. They find it a little bit aversive, and other people just say, oh my God, first time I used coke or meth, it was just like the best feeling I had in the world. So there is this continuum and spectrum of individual responses, which probably has a genetic contribution to it. And then in addition, as we all know, I mean, I shouldn't say we all know, but I think if you think about it, your relative position in society and what other forms of rewarding experiences you have influence your the chances of you developing a substance use disorder or an addiction. So while, I mean, I don't mean to lecture your audience, while it is absolutely true that you can be affluent and come from a loving family of high socioeconomic status with high educational attainment and become addicted. And we know famous Hollywood stars who have substance use problems or have died from their addictions. But it is also true that the prevalence of substance use disorders and addiction is much higher in individuals from lower socioeconomic strata. And the simple way we think about it is, for certain people, these substances, because of their ability to release dopamine in the reward circuitry, can be highly reinforcing, that is rewarding. And if you're living in a situation, an environment where there are no other sources of reward, there's no parks to play in to get reward from playing sports. The schools you go to are lousy. Your teachers are lousy. You're coming from a home environment where you're not getting, emotionally supported. You're not getting that kind of reward. If there's drugs in your environment, that's a major source of reinforcement, and you don't have that much other choice. So I hope it's this combination of your individual response to the drug, the environment in which you live in, and your access to the drug. I mean, this has nothing to do. These are, social issues. They're not neurobiological issues. So the type of addictions to substances that are most prominent in different countries and different societies vary enormously. So in Islamic society, where alcohol is not allowed, there is not an alcohol problem. There is an opiate problem, but there's not an alcohol problem. Whereas, in Western societies where alcohol is extremely available and is not socially prohibited, alcohol use disorder is a major problem. So probably more than you needed to know, but
no, this is great. This is great. There's also drugs, some of which you've studied that have little to no addiction liability. This includes the psychedelics. Some of them have little to no addiction liability. Some of them are even being studied right now as a way to potentially treat addiction to other substances. Before I want to preface this question with another little question or comment, which is right now there's kind of a battle going on in the literature over the term psychedelics and what that means. And so I'll let you define the term the way that you use it. But what is it about psychedelics, things like the serotonergic psychedelics like psilocybin or LSD, that make them different from classical drugs of abuse in terms of their addiction liability?
Yeah. So first, especially in the lay press, but even among my academic colleagues, I think, the term psychedelic is used very loosely. And most of us in the field would prefer to use more precise terms, because the group of drugs or substances that underlie the use of the term psychedelics actually have different mechanisms of action and different behavioral and psychological effects in human beings. So I think most lay people loosely, when they say psychedelics, they're mostly, but not entirely, referring to what I would define as classic hallucinogens, which are drugs like LSD, psilocybin, maybe mescaline, that have the common action that they activate certain subtypes of serotonin receptors. And as you know, serotonin is a major neuromodulator. Most of us in the field believe there's pretty good evidence that at least part of the hallucinogenic property is due to activation of serotonin 2A receptors. So when you really want to get really, more refined in your definition, you can actually talk about serotonin 2A hallucinogenics. So that's when I use the term psychedelics. But this, I grew up in the 60s and 70s, so I'm usually thinking of the classic hallucinogens, LSD, psilocybin, mescaline. People now include drugs like MDMA, which has gotten a lot of attention because of the application from Lycos to use it as a treatment for PTSD. I prefer to use the term for MDMA of an entactogen, because while there's a little bit of overlap with the effect it has in human beings, it's pretty, I think most people would say it's qualitatively different. And even though it's influencing serotonin mediated events in the brain, it mechanistically works in a pretty different way than classic hallucinogens.
Mhm.
Then people sometimes include ketamine as a psychedelic. I personally do not. Ketamine, those of us in the medical field define it as a dissociative anesthetic. So I think scientifically you have to use more defined terms, but even in the lay press, and as these agents become tested for their therapeutic efficacy, I do think it's useful to have use the term in a more precise way. Let's leave it at that. So then to answer your question, so I think the major reason classic hallucinogens like LSD, psilocybin, maybe mescaline, are not addictive in the classic sense is that they don't cause this massive release of dopamine.
Mm.
They're serotonergic agents. And in fact, I have not done this experiment myself, but and I have to actually check the literature. But I would predict and bet a lot of money that if you gave a mouse LSD or psilocybin and you measured increases in dopamine, you wouldn't detect much increase, maybe a little bit, but nothing like what you get with morphine or fentanyl or methamphetamine or cocaine.
Yeah, I'm pretty sure those experiments have been done, and the
Yeah.
answer is what you just said. It's little to no
Yeah.
extra
So, I
release.
mean, that's the excitement of studying these drugs neurobiologically is we can actually start understanding how they really work, and it actually makes some sense.
More recently, you've been studying MDMA and the
Mhm,
mechanisms by which it actually works
mhm.
to trigger its effects in the mammalian brain. You said MDMA is not a classic serotonergic psychedelic. It is a stimulant. I think it would be classified as a psychostimulant. What
Yeah,
is the addiction liability of MDMA, and how much is it tying into the dopamine reward system?
yeah, it's a great question, and there that's a little bit of a controversial issue. I would argue, but some of my colleagues in the field would disagree with this, that because it is an amphetamine derivative, by definition, it has addictive liability. I don't think it has as much addictive liability as the classic psychostimulants, methamphetamine, dextroamphetamine, cocaine. But it does cause, and here you have to I'm going to get a little technical. The MDMA that is being tested in clinical trials and the MDMA that presumably is used, it is an illegal Schedule 1 drug in the United States. So the one that is used illegally that has been used by millions of people at parties, at raves, is a racemic mixture which is composed of two different, we call them enantiomers, probably too much information. They're mirror images. It's
Yeah.
RS-MDMA.
Yeah, no, this is great because unlike ketamine, where I think there's quite a bit of not people do recognize that there's an S and an R isomer. I often find that people don't know this about MDMA, but
Yeah,
there's
so
two
the
isomers.
RS, and we have actually done studies testing this, as have others, and MDMA causes a massive release of serotonin in a way, because usually this, I'm going to anticipate a question, in a way that is dramatically different than traditional antidepressant drugs like Prozac,
Mm.
also which is called a specific serotonin reuptake inhibitor, SSRIs. And people often get confused on why does MDMA have this very profound pro-social effect, and some would argue empathogenic effect. And we actually just published a paper showing that in mice, MDMA actually enhances what I would argue is a behavioral measurement of empathy.
Hmm.
But I'm going off target here. So MDMA acts on the serotonin transporter and causes a massive release of serotonin. To a smaller degree, it also causes the release of dopamine to a much greater extent than classic hallucinogens like LSD, like psilocybin, which don't cause it increase. We have not done this directly, but my guess is MDMA doesn't cause as large or as rapid a release of dopamine as methamphetamine not sure about cocaine, but it certainly releases dopamine. And I mean, one of our papers was to try to dissociate what I would define as the pro-social effect of MDMA from what I would define as its addictive liability. And the hypothesis we came up with, based on our own work and other people's work in the field, is pretty simple and perhaps obvious, which is the addictive liability and its rewarding aspect, because at high doses MDMA is rewarding. People like it; they have fun on it. Mice like it. Is more due to the release of dopamine than the release of serotonin, whereas the release of serotonin in the same target structure really seems to be mediating its pro-social effects. And the reason I go into this level of detail is, while I certainly believe that MDMA, the RS form of MDMA, has therapeutic potential, especially as an adjunct to psychotherapy in the way it was used in these PTSD trials, I also do think it has some addictive liability. And if and when it gets approved in the United States by the FDA, I think there will be off-label use that may cause some individuals to have problems. And the reason I'm going to this level of detail is based on this potential addictive liability and the fact that MDMA comes in two flavors, R-MDMA and S-MDMA. And this is public. I'm not, you can just go to their websites. There are companies pursuing the R enantiomer of MDMA, R-MDMA, as a potentially better form of MDMA to use as a therapeutic agent. And the neurobiological basis of that is that it appears, more work needs to be done, that our MDMA preferentially releases serotonin more than dopamine, whereas S-MDMA probably, we have not tested this, this is the theory, has higher affinity for dopamine mechanisms, what's known as the dopamine transporter, and probably is releasing more dopamine than serotonin. So, enough said. So if it's the serotonin releasing properties of MDMA that is really accounting primarily, perhaps not solely, for its pro-social and perhaps its empathogenic actions, then the R-MDMA form enantiomer may have really good therapeutic potential with a higher safety margin, I think, way to put it.
So most of the MDMA out there, the stuff being used by Lykos Therapeutics for their PTSD trials, as well as what people are buying on the street for recreational use, is a mixture of the R and S isomer of MDMA, and therefore it's getting a mixture of these effects. I think what you're saying is there's some evidence that the R version of MDMA, the R isomer, is more tied to the serotonin release mechanisms and the prosocial effects, whereas the S one might be a little bit more for the heightened release of dopamine and might have a higher addiction liability.
That is the hypothesis. And again, the brain, unfortunately, is always more complicated than we want it to be. And, it will be very interesting to see as some of these companies begin testing our MDMA in clinical trials. Whether it's going to be in PTSD or in for other therapeutic purposes, it will be really interesting to see what happens. Because it may be you want a form of MDMA that releases a little bit of dopamine to get a little bit of that psychostimulant feeling while still causing the big release of serotonin to get what I would call the pro-social effect. And I will say, at least in mice, and whether this translates into human beings remains to be determined. My lab has generated a lot of evidence, which has been a little bit replicated by other labs, that it really is the release of serotonin in this target structure of the nucleus accumbens that, at least in mice, can account for its pro-social effects,
Hmm.
for its effect on wanting one mouse to hang out with another mouse in a non-aggressive fashion, for showing a behavior that we would say indicates that it's enhancing empathy in that mouse. But the proof will be when our MDMA form, enantiomer of MDMA, is actually tested in a phase 2 clinical trial, and we'll see whether it works or not.
Mm-hmm. So it sounds like, the racemic MDMA that's most common, the mixture of the two isomers, certainly has some addiction liability. It's non-zero, but probably not as much
just
as
to be clear, that's my personal opinion.
Okay.
I don't think you can argue that it has zero addiction liability. So
that would be surprising. Yeah,
that would be very surprising because of its actions on dopamine. Would be very surprising.
But we don't see people out in the real world behaving with respect to MDMA the way they do with fentanyl or methamphetamine.
So again, and that's why I like this way of conceptualizing psychoactive substances as having different levels of addiction liability, because in my view, that should influence how society regulates the access to these drugs. And that's why, for somebody like me, as a neurobiologist and a clinician psychiatrist, thinks it's important to really understand this and make these distinctions.
Mm-hmm. One of the things that people are really excited about with respect to psychedelics and related compounds, MDMA, ketamine, and so forth, is that there's some evidence that they can be therapeutically useful, and not just useful, but useful in ways that overcome a lot of the shortcomings that a lot of traditional psychiatric medications have. There's evidence that you can just give a small number of doses to see remission of things like PTSD, that you don't need to keep taking it chronically for months and years at a time. So they're fast acting in that sense. They also have the effect, or the side effect, depending on your perspective, of they induce subjective effects, hallucinations in the case of classical psychedelics, the stimulant and prosocial effects in the case of MDMA, the dissociative effects for things like ketamine. And there's some debate right now in the field in terms of whether or not the subjective effects are just epiphenomenal or they're required for the therapeutic benefit. Based on your whole career in synaptic plasticity and experience-dependent plasticity, how do you think about this?
I think that is perhaps the critical question in the whole field of psychedelic research, and in the context of using these drugs as therapeutic agents. Everybody in the field wants to know the answer to that question. My personal intuition is that it's going to be very difficult, if not impossible, but it's a personal opinion, not based on real data, to dissociate the subjective experience in response to these drugs from their therapeutic efficacy. That doesn't mean, so I really believe, and it's a little tricky. Do you need the trip for the therapeutic efficacy, or is it you can't dissociate them, but you actually don't need the trip? But I'm not explaining this very well. As you know, I'm sure there are companies, startup companies, biotechs, working really hard in developing analogs of these classic psychedelics, not only psilocybin, LSD, but including drugs like ibogaine, with the assumption that molecularly they can dissociate. They can make a drug that's, for example, still activates the serotonin 2A receptor, but does not cause a trip, or activates the 2A receptor. And maybe there was just a paper in Nature about the potential importance of a different subtype of serotonin receptor. As I said, my belief is it's going to be pretty difficult to separate the two. We're actually not going to really know the answer until these drugs that biotechs are creating are actually tested clinically. because we have very simple-minded models in mice to tell us whether a drug is hallucinogenic or not.
Mm-hmm.
But
we ultimately don't know until a human being takes it.
You really don't know till the human being, because the mouse can't tell you what it's experiencing. And let me just think, is there any clinical evidence out? Oh, the point I wanted to make is, so that doesn't mean, let's just say my intuition is reasonably correct. You still need some subjective experience to get the therapeutic action. That statement does not mean there is a lot of negatives there. There is not a lot of room to improve upon the classic hallucinogens in terms of, maybe you don't need to, I mean, currently in the clinical trials, let's say for psilocybin or even LSD, the doses that seem to have in early trials seem to have some therapeutic efficacy. You have the subjective experience, that is the trip, for 6 to 12 hours, which means you're sitting, the patient or subject is sitting there in a room with somebody sitting there with them in case they start having what's known as a bad trip. And that's a hard model for the clinical delivery of these substances.
Yeah.
So there's a lot of
scalable.
effort and companies working on, can you shorten the duration of the subjective experience and still get the same therapeutic efficacy? So as you probably know, there are hallucinogenic compounds such as DMT or I always forget, methoxy DMT, that cause a much shorter trip or subjective experience. And there are companies pursuing those as potential therapeutic agents. There are biotechs, again, figuring out ways of modifying these classic hallucinogens to shorten their duration of action. So there's a lot of room, even if the subjective experience is required, for maybe making these substances more user-friendly in terms of their therapeutic efficacy. But just to be clear, their therapeutic efficacy is far from firmly established. There's a few clinical trials out there that really suggest good effects, but how long the good effects last remains to be determined. And if you actually look at some of what I would view as the best done clinical trials, it doesn't cure everybody. It's still the case that there's a percentage of patients who don't respond in a therapeutically beneficial manner. So, I'm a believer that these substances absolutely should be studied, and they do have great therapeutic potential, but they need to be studied ethically and rigorously without an evangelical attitude towards them. That's just my personal view.
Mm-hmm. On the opposite side of the spectrum, when we think about the most addictive drugs, things like synthetic opioids like fentanyl, what makes them so addictive? Are they just causing the most dopamine release in those synapses we described earlier?
I know it sounds so simplistic, but that is probably the case. It's a little more complicated when we talk about the dopamine release. There's actually evidence from human brain imaging studies, a lot of it done by the director, not all of it, but some of it done by the director of NIDA, the National Institute on Drug Abuse, Nora Volkoff. And there's actually reasonably good evidence that the addictive liability of a drug correlates pretty strongly with two aspects of the dopamine release in this target structure, the nucleus accumbens. It's the magnitude of the dopamine release, but it's also the kinetics. How fast does that dopamine go up and how fast does it come down? And that's probably the case because it's not only the substance that makes it addictive, it's the route of administration. So one of the reasons smoking meth is actually more addictive than ingesting meth, because remember, a standard treatment for kids with ADHD is giving them amphetamine.
Yeah.
We're taking amphetamine orally. So that drug has to be absorbed by the GI system. Then it takes, I'm making this up, 10 to 30 minutes to get into the brain. And the kinetics of the drug delivery, let's say an amphetamine taken orally, is it gradually gets into the brain, gradually decays from the brain. So the effect on dopamine levels is very gradual increase and a very gradual decrease. If you smoke or inject methamphetamine, it gets into the brain instantaneously, especially when you smoke it. And I don't know if you're old enough to remember the crack cocaine epidemic. And the reason crack cocaine was so addictive is the route of administration. People were smoking it. It gets into the brain instantaneously. It causes a very rapid, very large increase in dopamine, which dissipates, which disappears over the course of minutes. And that's why if you talk to people who really were smoking meth or crack cocaine, as you know, as a neuroscientist, there's this famous experiment done by James Olds showing that if you put a wire into an animal's reward circuitry and you allow it to press a bar to activate electrically the reward circuitry, the animal will just sit there and repetitively press the bar for hours on end. And, smoking crack cocaine or methamphetamine is as close to an electrical stimulation of the reward circuitry as you possibly can get. So you get this very rapid release, this jolt of dopamine, this highly reinforcing event, then it's gone 2 to 8 minutes later. And then what the individual experiencing this is then left with this overwhelming craving or wanting of this drug. And it's a very powerful intermittent reinforcer. And just to go off on a complete tangent here, the gambling industry has paid a lot of attention to the neurobiology of addiction and the neurobiology of compulsive reward seeking. So it's not an accident that, slot machines on a Vegas casino floor, they deliver rewards on a partial reinforcement schedule to make people, I hope this is making sense. So
they're engineered based on our neurobiological knowledge to give rewards on exactly the most reinforcing schedule.
And so, even so, I imagine with a drug like fentanyl, I'm not sure if the imaging studies have been done, but because it's such a powerful opioid, and I think a lot of people smoke it. I believe that's a pretty common route of
administration. As far as I can tell in Seattle, they appear to be doing that.
So it's just like smoking methamphetamine and maybe even worse. So it's getting into the brain rapidly. So I really think it might be, of course, other things are going on, but it really might be that simple. And it's a little frustrating because we kind of understand the underlying neurobiological mechanism, but that still hasn't allowed us to develop, a treatment that is as effective as we would like.
Mm-hmm. As a psychiatrist and a synaptic plasticity expert, how, thinking about amphetamine, which, goes by the name of Adderall, when we think about the ADHD medication, how concerned are you that we are giving so many young children at such a young age a psychostimulant like that?
I don't know the clinical literature. I'm concerned, but modestly concerned because, I always say, drugs are not inherently good or bad. They're drugs. And every drug, even aspirin or what are known as NSAIDs, drugs like ibuprofen or, Aleve or naproxen, every drug has most drugs have some positive aspects to them and some negative aspects. So in the case of giving Adderall to kids with ADHD, the benefit is it really does help some of these kids focus, function better in school, socially interact in a more adaptive way. And that's really important, because if you're not able to function in school, you're not able to socially interact when you're in elementary school or junior high school, that is going to have an impact on the rest of your life. So so I want to make it clear, these are not, there's a lot of benefit, but it's balanced by, there is some risk profile that a certain percentage of these kids are getting exposed to a drug with addictive liability. And I actually don't know the literature. I think there is a higher, I may be wrong on this, a higher like prevalence of subsequent substance use problems in kids who were treated with Adderall early on. But I seem to remember seeing some of that data. I don't think it's profound, though. So I hope I'm answering questions. So it's a balance, I think. If you're a subscribing physician, you just want to be thoughtful and careful and balance the risk versus the benefits, and, talk to the parent of the kid and make sure there's an understanding.
Earlier, we briefly mentioned the role of endocannabinoids in certain forms of synaptic plasticity that are really interesting and fairly well studied. Then, understanding that we have a endocannabinoid system that ties into some of these plasticity mechanisms, it's not a big leap to imagine that that's why things like THC, these exogenous cannabinoids that some people consume, affect learning and memory.
Yep.
On the addiction question, in terms of how exogenous cannabinoids like THC work, are they tapping into the dopamine reward circuitry, and what is their addiction liability? Because you see the full spectrum of opinions out there about marijuana.
I think cannabis products do have a addiction liability. I do think it's much less than opioids or psychostimulants. I think there it's actually useful to not really to talk about cannabis use disorder, because in general, and then I'll answer your dopamine question in a second. And I'm making gross generalizations here. Obviously, I know from, I grew up in the 60s and 70s. There are millions of people who can use cannabis products in a way that they use alcohol with no significant damage done, meaning just use it kind of socially on occasion, and it's really not doing any disruptive things to their life. There's also certainly the case that a percentage of cannabis users do develop a cannabis use disorder, and whether that's 5% of users or 15% of users, I am not sure, but it's not zero. It's a little bit different in the sense that usually these individuals kind of develop, for lack of a better term, an amotivational syndrome, where they're just, they're not motivated to work as hard. They develop some cognitive issues, some learning and memory issues. They're in general, and it's interesting, and it has to do with the other actions of the drugs. They're not craving the drug quite to the same degree, in the sense that you don't often hear about people committing violent crimes because of their cannabis use, or getting in, automobile accidents I'm not going to even comment on that. I'll bet you there are a lot of automobile accidents due to the use of cannabis, but violent crimes really aren't associated. And that's because of the other effects of cannabis, of marijuana and stuff. You're not, you're chilled out. You're just sort of, whatever you're high, and you're not really, and you're not particularly aggressive or violent. So in terms of the dopamine, I should know this, I don't. I think it does cause some dopamine release. I think animals like mice can be taught to self-administer it, but I'm pretty confident if you gave a choice to, let's say, a mouse or a rabbit or a rat, and they could self-administer amphetamine versus THC, or an opioid like morphine or fentanyl, they would always choose the former, the substance. So I hope I'm answering your question.
Mhm.
So
So
again,
again, it sounds like this is a drug where there's definitely some addiction liability, and it's certainly not zero, but it's not going to be as high as the things that we normally consider to be the most
absolutely. I
highly
mean, put it
addictive
this way:
drugs.
I think the addiction liability of alcohol and nicotine are substantially higher than cannabis. That doesn't mean cannabis is zero.
Mhm.
Um, but and we're going to see this as cannabis products become more and more legalized, at least in the United States. There is going to be, you're going to see more and more people with cannabis use disorders. I predict it's never going to be as high as the number of people who we used to call alcoholic, and now we say have an alcohol use disorder. And I don't think it will ever be as high as people with a tobacco problem, with a cigarette problem. And that, again, I think can actually really be explained by the neurobiological actions of nicotine, alcohol versus, as you pointed out, the psychoactive substance in cannabis, which is THC.
Is there anything that you want to reiterate for people, or any final thoughts you want to leave people with about synaptic plasticity or addiction and the topics that we discussed today?
Not really. Synaptic plasticity is um, a group of phenomena in the brain that makes the brain different than the hardware of a computer. It's why our brains are constantly changing from the day we're born to the day we die. I mean, your listeners, their brains, their physical properties of their brain, the biochemical processes going on in their cells and their synapses have changed as a consequence of listening to this podcast. Um, and the hardware of a computer doesn't do that. So, but I would also, remind the listeners that there are adaptive forms of plasticity, the types of plasticity that we use for learning and memory. We use to, learning and memory that allow us to remember bad events in our life, good events. But there are also maladaptive forms. So one thing to just gently point out to your listeners is there's a lot of excitement about psychedelics being special plasticity promoting agents. And I think that is probably the case, that they can promote different types of plasticity in the brain very rapidly. But I would also gently remind them that by that definition, cocaine and opioids are also plasticity promoting agents. They just promote bad forms of plasticity by definition. So it's a little simplistic to talk about psychedelics as psychotoplastinogens, which is a very popular term, but because a little bit by that definition, cocaine, fentanyl, morphine, alcohol are also psychoplastinogens. So again, everything in the brain is not simple. You have to be able to think about these phenomenon in a little bit more sophisticated way. And that applies not only to our understanding of psychedelics and synaptic plasticity. Now I'm going to get a little political here. It also applies to how we think about societal problems in the United States. So I encourage your listeners to have a sophisticated view of how to solve societal problems, including addiction. That's it.
All right. Well, Dr. Robert Malenka, thank you very much for your time. That was fascinating. We covered a lot of interesting stuff, and I think people are really going to get a lot out of this one.
Well, I hope I explained things adequately, and it was a lot of fun to talk to you. So thanks for having me.